মানে ইউআর লাইভ দেখাচ্ছে, কিন্তু আমার ক্যামেরাটা অন হয়নি। দেখুন আমার এটা অন হয়ে গেছে। হ্যাঁ, কিন্তু আমার ক্যামেরাটা অন হয়নি। না, ল্যাপটপ।
কথা বলো।
হ্যালো, দাঁড়া দাঁড়া করছি।
হ্যালো,
দাঁড়া, একটু কাটবেন না, ধরে রাখবেন আমার। হ্যালো হ্যালো, ঠিক আছে।
তুমি কোথায়? হ্যালো, হ্যালো। আর কতক্ষণ লাগবে?
হ্যালো 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 হ্যালো।
হ্যালো, কেটে গেল। ফোনটা কি কথা বলি না?
হ্যাঁ, সাউন্ডটা দেখলাম আসছে।
Good evening, everybody. So, due to some technical issue, you cannot see me, but I'm trying my best to explain you this new subject, that is the biopsychology. So uh, this biopsychology, it is BPC C102, and this is your second paper, right? So this biopsychology, it uh, it is divided into four block here. And the first block, it covers the introduction to biopsychology. That means what is biopsychology, what are the methods, and then the next chapter is the next unit. That means these blocks have two units each, right? So that means these units are like divided into two, that is the introduction to biopsychology and the neurons and nerve impulse, and this is under the block 1. And the block 2, it has the same, like the similar way, it also has two parts, that is the central nervous system, that is the unit 3, and the unit 4, it is the peripheral nervous system. Now the block 3, it is the it is a very important block of this paper, because it is the brain lateralization. It covers the whole chapter, or the whole unit, that is the unit 5. It covers the hemispheric specialization. And the block 4, it covers the glands and the behavior. That means all the endocrinal system, the activities, the functions, everything. So this is all about this content of this biopsychology. Now, first, today I will start with the introduction of the biopsychology. Now, what is biopsychology? I told you that in this paper we will mostly covers the biological part and the relation between the biology and the psychology together. Now, in this unit 1, that is the introduction to biopsychology, here we will start with the definition part, that is what is biopsychology. Now, if you see the study material that is given from the Ignou office, you can easily find that first of all you have to start with the brain. That means the relation with the biology and the behavioral issues that are together. That means the biology plus the psychology. So the relation between the what this field mainly covers, the relationship between these two. So it is also known as the psychobiology, the behavioral biology, and as well as the behavioral neuroscience. So this is all about the meaning of this biopsychology term. Now, the nature and scope of biopsychology, from there we will start with the we will know, as we have already studied in the other papers, that this biological approach to the study of humans and the animal behavior together is the part of this biopsychology. Now, this biopsychology started as a separate discipline of this neuroscience, uh, mostly after the 20th century, when we will find the D.O. Hepps. He was a Canadian psychologist. Um, Through his different publications, like the Organization of Behavior in 1949, and also the field of psychology and neuroscience. After that, in 1949, there were other robotics and other fields of engineering and as well as psychology together with the neuroscience. This biopsychology came into existence as a separate unit of psychology. Now, this biopsychology, as you know, that it is a relationship between the biology and the psychology. There are some other terms like the neuroanatomy, that means the structure or the study of the whole structure of the nervous system together. So that is, it is termed as the neuro, that means the neural or the nervous system, and the anatomy together, that is the neuroanatomy. Then the chemical aspects of the nervous activities together, that is the neurochemistry. Then the neuropathology, that means it is also another study of disorders of the nervous system. That means what are the disorders, how to treat these disorders, etc. Now again, another part is the endocrinology. That means the relationship between the neurons or the nervous system with the endocrinal system. Now endocrine system, that means the glands, that is the thyroid glands, the gonads, etc. There were other some endocrine glands that we already know. Now this neuroendocrinology, it is another study, and it deals with the interaction between the nervous system and the endocrine system. Now this neuropharmacology, it is another study, that means it mostly deals with the impact of the drugs on the neural activity. That means due to some disorders we are taking some drugs, and the activity of that drugs, that means the neuro, the nervous system, and the relationship between the pharmacology together, that is the neuropharmacology. And the another part is the neurophysiology. Now neurophysiology, that means from the term physiology we know that it is the functions, it actually studies the functions, the activities of the nervous system, right? So these are the introductory part of this biopsychology. Now there were various other researches. There were some researches in India, like the Council of Medical Research, that is the ICMR, and there were some other various researches on this biopsychology. And the research in this biopsychology may be a pure research or applied research, and there were various empirical investigations with the applications of various experiments on this field. And this biopsychology, later on, as you all know, you have already learned about the Pavlov's experiment of dog. Hmm. So you all know that Pavlov, he was a Russian physiologist, and his introduction to psychology, that is a referred, it was referred to classical conditioning. You have already studied this in, I think, in learning chapter. So this Pavlov was a Nobel Prize winner in 1904 for his research on this physiology of digestion. So his famous experiment was on dog. Hmm. So that was also a relationship between the physiological aspects, that means the formation of saliva, the digestive system, with the physiology. So there were some other applied research as well as pure research in this field. Now, the divisions in biopsychology, as I told you, there were some important divisions like the physiological psychology. Now, it actually means the physiological psychology, that is the controlled experiment conditions, how the brain acts, or the stimulation of the brain, and also the effects of the behavior. So that is the main part of this physiological psychology. Now this psychopharmacology, that means the uses of these drugs, that means pharmacology means the therapeutic drugs that we are taking, that part, that means the psychopharmacologist, they actually use the drugs to stimulate any types of neural systems, and then observes that what are the effects of the behavior. That means the problem of the nervous system, or if there is any disturbances in the nervous system, they can be controlled by this pharmacological experiments, right? Now then comes the neuropsychology. The neuropsychologist, they actually study the patients who have had some head injury, or they have some, due to some accidents or due to some traumatic sufferings, they have some brain damage or maybe brain tumor, etc. Now there, this neuropsychological test, they actually helps to diagnose the patient, mostly for the treatment purpose. And this subdiscipline actually specially uses this case studies and quasi experimental studies. If there is any type of neurosurgery or any type of brain damage, etc. Now this neuropsychology, after this neuropsychology, the third one that I will tell you, that is the psychophysiology. Now this psychophysiology, they actually, the psychophysiologists, they use this non-invasive technique. That means like EEG. What is EEG? That is a electroencephalogram. Any types of it actually see that if there is any type of problem, like mainly if attention deficit due to some, or due to some learning problem or memory or emotions, etc., these type of problems can be detected by this type of test. And these tests, namely the EEG, that is the electroencephalogram. Then there are some other tests like galvanic skin response, that is the GSR, etc. Then heart rate, then blood pressure, pupil dilation, etc. So all of this can be measured by these type of tests. Now after this psychophysiology, the next part is the cognitive neuroscience. Now this cognitive neuroscience, it actually studies the neural basis of this cognition and the higher order in cognitive processes. Now what are the higher order cognitive processes? I think you already know, like the thinking, the perception, the memory, the attention, etc. So these are the cognitive, higher order cognitive processes. Now these type of cognitive neuroscience, that means if there is any problem in thinking, in memory, in perception, in attention, or lack of concentration, or memory problem, etc. So this can be detected by this new field, that is the study of the neural basis of cognition and the higher order cognitive processes. Now, after this cognitive neuroscience comes the comparative psychology. Now, from the term itself, you know that comparative psychologists, they actually study the behavioral aspect of the different species to understand that what type of behavior that a living thing will have, like from the evolutionary change, from genetic or adaptation perspective, etc. Now there are some ethological researches on this comparative psychology. So I told you from the beginning, I'm again telling you again, so that you can just visualize that the divisions in biopsychology, there are physiological psychology, number one; number two, the psychopharmacology; number three, the neuropsychology; then the psychophysiology; then comes the cognitive neuroscience; and lastly, the comparative psychology. So, from the term itself, you can easily define that what type of psychology it is. Now, after this comes the methods to study this biopsychology. Obviously, there are some fixed methods to study this biopsychology. Now, what are they? Before we start the method, first I will tell you that about Paul Broca. He discovered this that there are some regions of brain which are actually responsible for our language. So he was a French neurologist, and in 1861 he found that one of his patients who had lost the ability to speak had damage in left frontal cortex. Now, while I will explain you the different parts of the brain, I will come to all the areas, all the structures of brain. Now, that is the left frontal cortex. There are right frontal cortex as well as the left frontal cortex. So, he found that if there are any damage in the left frontal cortex, then there must be some problem or loss of ability to speak. So, according to his name, this type of problem is known as Broca's problem. So, later on, we will study. Now, after this, we will study the first method, that is the ablation method. Now, this ablation, from the term, it means the removal of any brain area. That means just by the surgical instrument. Okay. So this ablation method, it involves the destruction of some tissues in the brain, obviously. That means to separate out the areas of problem which are the functions of which are making a problem in that region. So destruction of that tissue from the brain. So that is the ablation method. Now there are some techniques of this ablation method. Now the main techniques are the aspiration, the radio frequency, the knife cuts, the cryogenic blockades, and the nerve poison. Now this aspiration, it is a fine thin needle like thing, and it is placed on the tissue, and from there the surgeon can access the instruments that needs to be examined, and it is aspirated and the contents are then placed on the slide and examined, or the diagnosis starts, or from it the analysis of the problem can be done. So this is another technique of this ablation method. Now, the second technique that is discussed is the radio frequency. Now, radio frequency, it is often abbreviated as RF. That is, a current is used to produce the subcortical lessons, and the wire is placed on the tissue which needs to be examined. Now, why it is known as radio frequency? Because there is a high frequency current in between, and that is alternating current. So the duration and the intensity of the current will determine the problem or the size or the actual position of the lesson. So this is all about this radio frequency. Now, number 3 technique of this ablation is the knife cut. Now, what is this knife cut? This knife cut means just cutting that region. That means sectioning. It is used to make cut on any area of the nerves of the brain, and there is a special device that is used to make this subcortical knife cuts. So this is the knife cuts. From the term itself, you can easily know that what is knife cut. And the next technique of this ablation method is the cryogenic blockade. Now, what is this cryogenic blockade? This is a temporary way of inducing this lesson. Now, why temporary? Because this cryo probe is placed in that position of the brain. Okay. Now, through this tube, a coolant is sent in this problem area to study that what type of problem is there. Now, what is the function of this coolant? It actually reduces the temperature. So, what is the function of a coolant? Obviously, it will reduce the temperature. The same thing happens here, and as it's steep to such an extent that the neurons stop sending the impulses. Now, since this temperature is maintained at above freezing point, that means 0 degree, there is no structural damage. Okay, so this is what this cryogenic blockade means. So, just sending the coolant in the particular region of the brain. Okay, then comes the last technique of ablation, that is the nerve poison. This nerve poison, it is the chemical, there is some particular chemicals such as asconite acid or abiotenic acid are used as the nerve poison. Now to create, now what are the function of these two poisons? They actually create the lessons in a particular area of the brain where there is a particular problem, and the destruction is only partial, that is only the cell bodies are destroyed here. So these are the a few techniques described, that is the ablation methods, like the aspiration that I told you, the radio frequency, then the knife cut, the cryogenic blockade, then comes the nerve poison. Now, histological method, it is the second important method of this biopsychology. Now, here, with the advent of the technology, this type of method was applied to study mostly the anatomy and the functions of the human brain. Now, for studying minute details of tissue, the microscopic investigation or histological methods are mainly used here. Now, again, there are some various types of these histological methods. What are they? Number one, it is the fixation. Now, in this method, this fixative is used to fix any type of tissues. That means just like a sticky fixative. Okay, so this tissue is cleaned and dipped in a fixative solution, and the most commonly used fixative is formalin. I think it is known to you, formalin, and this is used as a fixative, and this fixative prevents actually the autolysis and makes the tissue hard. So that particular region of the tissue becomes hard. And what happens? The tissue is not so hard to be cut using a microtome. So what happens? The brain tissue is freezed so that the tissue becomes more hard. And it is also used to stabilize the neural tissue. So it is another most important type of historical method where they are using some fixative like formalin to fixate the tissues in that particular region of the brain. Then comes the uh, freezing. Now, freezing, that means the tissue needs to be frozen in order to be sliced. And how it is done? It is done by dipping that region of the tissue into some sugar solution and then put in a freezer. So this is the method of this freezing. Then comes embedding. Now embedding means, you all know that, covering that particular tissue by some paraffin like material or nitrocellulose, and by covering that, that region is observed through the microscope, and it is just like the albumin of the egg. Okay, so the slide is dried, it is then dyed using different chemical solutions. So this is the embedding technique of this method, that is the histological method. And then comes the staining. Staining means just putting some colors, like stain colors by the myelin sheath to study the nerve path. That means now which way the nerve is functioning. And it also stains the membranes of the Golgi Cox stain. Now, in the next unit, when I will explain the structure and functions of the neuron, I will just go through all the parts of the neuron, the Golgi body, the everything, the swan shell, the myelin sheath. So, by coloring this myelin sheath, this type of staining procedure is done. The third important method of this biopsychology is the psychophysiological recording method. And it is very often, you all know that what is EEG. You already heard these terms, I think. So this electroencephalography, that is the capital E, capital G, and it is the electrical, by this method we can easily observe that what are the electrical activities of the brain, and it is recorded using some electroencephalography. That is a machine, and the techniques is the electroencephalography. The graph is the machine, that is we can observe, we can easily see the recorded activities of the brain, and the techniques are used, it is known as electroencephalography. Now these electrodes, they are placed on the scalp of the patient. If you see, there is a picture in your study material. So these electrodes are placed in the skull of the patient, and through the computer they can easily see, or it is displayed in a screen, that how the brain is working. The functional procedures are all displayed in the screen, and we can easily take a printout from that, and we can see, and in what type of brain disorders we actually use this EEG. Say for example, any brain damage, or any cerebral pathology, or after some stroke, or if there is any epilepsy or seizure, or any type of brain problem. Hmm. Now this after this EEG, there is another techniques that is used, that is magnetoencephalography, that is MEG, and it actually measures the changes in the magnetic field on the surface of the scalp, and produced by changes in the basic pattern of the neural activity. Now, after this EEG, this magnetoencephalography, the electromyography is another type of technique, that is EMG. Now, myography means it actually record the muscle tensions, and with the help of this electromyogram, that is the EMG. Now the electrodes are attached to the skin over the muscles, that is to be investigated. Now when the muscle contracts, producing this muscle tension, it is recorded. And this information helps to know that if there is any problem in the muscle tone or the functions of the muscles. Now, after this technique, there is another important technique, that is EOG. That means electroculography. Now, while you will study all this, always try to remember by writing these techniques. As this from the term, you can easily see that electroculography means the eye movement. It actually records what type of eye movements, and by using this electrokilogram, that is the EOG, and there is a slight difference in the electrical potential in the back and the front portion of the eyeball. So these are recorded by this electroculography. Then electrodermal activity means skin, the skin conductance level, that is the SCL. So these are recorded. The main two indices of this electrodermal activities are the skin conductance level and the skin conductance response. So you all know that what is response and stimuli, as you have already learned in the previous papers. So this response means how the skin conductance actually responds. This electrodermal activity of the skin is recorded, and by attaching the electrodes on the figure, we actually can record the fluctuations of the skin conductance. So obviously these fluctuations helps us to understand if there is any type of change, and the behavioral, the emotional change, like the sweat gland, the secretion of sweat glands, etc. These can be easily observed by this electrodermal activity. Now this electrical stimulation, it is another most important, or the brain can be actually stimulated this electrically to gain some information about the working of its various neural system. Now what are the working of the various neural system? So how we behave, what are the typical behavior of any emotions, etc. So how these can be recorded? These recordings of the electrical signals are then displayed by some various methods. They are the by the help of this oscilloscope, or oscilloscope is nothing but the ink writing oscilloscope, where we can see the recordings per unit time. Now this oscilloscope, through this oscilloscope, there is a display of electrical recording where the cathode ray tube is displayed, and this record the activities of the single neuron. That means just imagine there are innumerable neurons, and the single neuron, how it the activity of a single neuron can be easily seen or easily observed by the recordings. Now then again comes the ink writing oscilloscope. What is this ink writing? Like, it is also the another name of this ink writing oscilloscope is the polygram. Now this polygram, the evoked potentials are recorded with the help of various electrodes that are attached to the scalp. And there are some needles or pen tips, sharp tips, and which record the neural activity, and there is the recording is done in a rolling paper with the ink tip, and we can easily see that the recordings, and we can find if there is any type of problem in the brain, mainly due to if there is any brain damage due to some epilepsy, some seizures, or due to some brain tumor, etc. Then obviously you all know computer is another most important technique, where we can easily with the help of the computer, it actually displays or stores the information. If there is any type of problem due to the epilepsy or during the time of seizure, then it can be easily recorded, and we can compare it by the normal situations. Now there are some chemical stimulations which are also used, like some chemical stimulant which actually helps to stimulate the brain and make these lessons. And these chemical stimulants are nothing but the drugs that are used. Now There are some important techniques like the 2-deoxyglucose technique, that is known as 2DG. And this 2DG is a radioactive drug. It is often used, and it is actually injected in the animal, and the researcher waits till it is absorbed by the brain. That means it takes time to reach the brain, and thereafter the animal is killed and the brain is removed and slices are made, and this auto radiography is conducted. But in case of humans it is not done, obviously. So then another useful technique is cerebral dialysis. You often heard this term dialysis. The If any patient has any problem in kidney, so often this dialysis we already know. But this is the cerebral dialysis. It is another useful technique that is used, and there is a tube is inserted into the animal's brain, and from this tube we actually can record or pass the neurochemicals released through the chromatograph. And it is obviously the involuntary. In another techniques also, there are some recordings are done by releasing some neurochemicals released in the brain of that particular animal, particular region of the brain. And there is some change. It is actually compared with the normal situation, and we can easily find if there is any problem in that particular area. Then comes the stereotaxic lesson. It is another important type of lesson, and we have already studied in the ablation method that um, it is when ablation is not possible, if there is any problem in doing this ablation, then this type of technique is done. And researchers use these stereotaxic techniques to damage a structure in the interior of the brain. And by damaging that region, they actually compared the situation with the normal person, and see that if by the help of this stereotactic atlas, it helps to locate the exact point of the brain surgery or what happened in that particular area. So this is another very important type of lesson, that is a stereotaxic lesson. And neuroimaging is all, you I think you already know that the X-ray technique is the neuroimaging. It is a very important method. We can easily observe the function, and if there is any type of injury or brain damage, it are these are clearly observed by this neuroimaging techniques. Now, the most important neuroimaging technique is the X-ray technique. That means this technique is very common, and the conventional X-ray did not help much in this observing this neural activity. So the cerebral ventricular system and the circulatory system within the brain is examined by using the contrast X-ray technique. And in this technique, there is a substance is again injected into one compartment of the body, and it actually takes some times to absorb this type of X-ray. And after that, this X-ray, either less than or more than the surroundings tissues, it actually absorbs. And after that, we can take or see the contrast region, the X-ray photo, by the help of this X-ray photography. This X-ray method is very important. After that, the most important scan that that is the CT scan, the computerized tomography. Mm. So this CT scan, it is also another type of X-ray technique, and it is actually with the help of the computer we can easily view the structures of the brain, the functional part, and the other all the other particular regions that are involved in any type of problem in the brain. And the x-ray is passed from one side and the x-ray detector is placed on the other side of the patient's head. That means if, the left and right. And slowly this x-ray and the detector are rotated and the photographs are taken from all angles like left, right, back, front, etc. So that means the three-dimensional picture of the brain is observed by this computerized tomography, that is CT scan. Then comes the MRI. It is also well-known type of neuroimaging techniques. And this MRI, it is a magnetic resonance imaging. And this technique is used when detailed pictures of the patient's brain is to be investigated. And this technique uses a strong magnetic field to gather images. And this MRI is there is a particular chemical is injected, and this technique helps to know the oxygen in the blood and the flow of blood in different brain, or what are the activities that are responsible for that blood flow. And all of these are observed by this MRI techniques. So it is a very important neuroimaging technique. Now the next one is the positron emission tomography, that is the PET scan. It is also known as PET scan, and it is a very very important and costly neuroimaging technique. So this PET scan, it helps to detect there is if there is any changes in the metabolic activity, or if there is any abnormalities in the brain functioning. It does not provide any structural information about the brain, but the functional or the activities are recorded here. And this it is an invasive method. Again, there is an injection given to the patient of the radioactive 2-deoxyglucose, and that is, it is known as 2DG, is given to the patient through the injection. And it takes 1 or 1.5 hours after this injection to absorb this 2-deoxyglucose. And after that, this recording starts. So this images, it actually records all the functioning or the activity parts of the brain. It is mostly used for cancer patients, where this it can detect that if there is any type of problems in the functional part of the regions of the nervous system and the brain. Then the functional MRI, it is another type of MRI, and this small f and MRI, it is less expensive than original MRI that you have already studied, and it is also less risky technique. Now these techniques are obviously risky because there are some injections of some chemicals. Like in PET scan, there is an injection of I told you that 2-deoxyglucose, and it makes the temperature of the body high. And obviously if there is any problem in the patient, then sometimes after this test, there are some functional problem to the patient. Maybe if that patient is going through any cancerous or malignancy. And so this small fMRI, it is less expensive, less risky technique than PET scan. And this fMRI, it's primarily measures the blood flow, that is a hemodynamic response of the body, and also the cognitive neuroscience, any problem or any procedures that are going on in the neural and as well as the brain, nervous system as well as the brain. Now another technique is the diffusion tensor imaging, that is known as the DTI. and it is a method often used. It actually indicates the pathways along with water diffuses. Um, and this water diffuses in the tracks. There is a bundles of axons, and this diffusion tensor imaging gives image of this major tract, and this DTI, it actually determines the location and also the orientation of the neural axons in the brain. Then after this diffusion tensor imaging, the next one is the transcranial magnetic stimulation, that is a TMS, and it is a most important technique. It turns off the part of cortex that I told you in the beginning, the cerebral cortex, and by creating a magnetic field, and it is temporarily inactivates the neurons below the magnet. So the mostly this temporary inactivities are done by this type of method, and then the researchers use this TMS also in mapping this technique. And here the TMS coil is placed on the motor regions of the cerebral cortex and stimulating different regions along this cortical strip. Then the muscle movements are also observed by this. So this is another important technique, that is the TMS or the transcranial magnetic stimulation. Now, after this, the next type of methods is the assessment part. How this is done? This is done by some techniques or some tests to assess any type of cognitive problems, maybe cognitive functioning, maybe learning difficulties, maybe memory damage, maybe any intelligence problem, language, maybe language problem, or any type of decision making, etc. Now, for this, there are also some important test that is well known. Wechsler's intelligence scale, that is the WAIS. It is very important because um, it is very important because from this type of, by applying this type of test, we can easily see or easily observe that if there is any problem in the intelligence area, that means the intelligent quotient. So when you will study the intelligence chapter, then you will see that what is intelligent quotient, how you can easily see the intelligence of a child or an adult. So by taking the help of this adult Wechsler's intelligence scale, that is often known as WAIS-3, there is some fixed intelligence scale for the adults and also for the children's, and the preschool children, the primary children, then 6 to 16 age group. There are some different intelligence scales to detect the intelligence of the school children or as well as the adults. Then the next assessment technique is the Halstead Written Neuropsychological Test Battery, and it is named after Written in 1986. And this battery, it actually consists of a set of neuropsychological measures, and often some trained psychologist or some clinical psychologist, they actually take this type of test to assess if there is any major cognitive problems, any sensory issues, or any type of motor functional difficulties. And there are some fixed test batteries for the adults, the older age group, both. Then comes another assessment technique, that is the Luria Nebraska Neuropsychological Battery. It is also a standardized battery, and it came into existence in 1986. It is a very important neuropsychological test. It is used to diagnose if there is any brain damage, if there is any brain dysfunction of adults, adolescents, as well as the older adults, etc. That means For all the age groups, there are some fixed test batteries to assess this type of intelligent functioning. Then the re-ostric complex figure test. This is another type of tests for assessment, and this is also named after Ray in 1941, and it actually helps to assess the visual perception of the patient or the visual memory in the brain damaged individual, etc. It is named after two persons, that is Ray and Ostreth in 1944 and Ray in 1941. Together, they developed this complex figure test, and also it is an important test to assess the intelligence of the children, that means 4 to 15 years as well as 16 to 60 years of age. Then Delhi's Kaplan executive function system, that is another important assessment technique, and it is a neuropsychological assessment technique. It actually helps to execute or the functioning part of the thinking procedure, that means the cognitive area, inhibition of anything, or it helps in solving any types of difficult problems, or planning, execution, then impulse control, concept formation, creativity, as well as abstract thinking. So this is a very important or elaborate cognitive functioning. It actually can detect or assess these all cognitive processes, and it is named after Dellis and Kaplan and Kramer in 2001, and it may be administered and it is standardized measures to assess executive functions, and also these tests are for the children as well as the adults. Then comes the digit span test. Digit span test is another type of test that is also known as WAIS, and the aim of the test is to measure what type of working part or working memory of the individual, or if there is any impairment on waking memory. Waking memory means the memory of today, like what uh, food we have taken in the lunch or just now. So that is the impairment of the waking memory. So it can be assessed by this digit span test by applying this. Then Wisconsin card sorting test. It is another important test where the patient has to sort some cards by shifting the cards, and it actually assess the if there is any problem on the frontal lobe or dysfunction in some frontal lobe. And here the if the patient has some brain injury or some psychotic disorders like the schizophrenia or any type of mental illnesses, so there are some 64 cards, and the patient has to match these cards according to the numbers and figures, and there is a fixed rule by observing that we can easily assess the problem if there is any problem in the brain. Now then comes the dichotic listening test. Now from the word you already know, di means two and otic means ear. That means the test is a non-invasive test, and initially it used to investigate the selective attention. That means the sound from the left ear as well as the sound from the right ear, and it can be easily observed by the standard this dichotic listening task, and the participant or the patient is simultaneously presented with two different auditory stimuli through the earphones, that means from the right earphone as well as from the left earphone. And from this we can easily assess if there is any type of problem, auditory problem. It can be easily detected. Now there are some research ethics in this biopsychology. Now what are these research ethics? These are nothing but we actually cannot see, or there are some various ethical issues that need to be taken care of while doing this research in biopsychology. Why? Because we cannot use some coolants or any type of injection that can easily damage the brain, or the patient can die, sometimes may die. So we cannot use this type of things for the research purposes. And because there may be some problem, so when human participants are selected, then the consent is taken from the participant, and the doctors that actually treats them, the therapist and the caregivers, etc. So the ethical part is the most important part. And if these experiments are done on rats or animals, then also we cannot use this type of techniques. If that animal die after this injection, that obviously this type of research or experiments cannot be done. So these are the research ethics in this biopsychology. So by this today, up to this, due to some technical problem, you cannot see me today. Sorry for the technical issue. I am Dr. Ruma Mukherjee. So I will teach you this biopsychology. The next class, I think you will be able to see me. So sorry for it. Now, if there is any question regarding this unit, then the next class, before the class, I will try to give your answer. And you please go through this unit. This is a very important unit, and it easily you can get marks from this unit or in this paper, because this is very much related with the life science or the biology, as the relationship with the biology and the psychology, by some methods, by studying the functions and the structures of the brain, the nervous system, etc. We will go through these units, and obviously it is very important and interesting too. So today, up to this, thank you everybody. Go through this unit, and if there is any problem, please let me know. I will try to answer you. So, okay, thank you. Good night.